Okay, thank you very much. I would like to just mention that I come from a region that is at the moment under great stress because of the refugee crisis in Europe, and maybe many of you have heard, so I just want to point out where we are. It is just right in the center of Europe here. The refugees actually have taken this route. Instead of going to Hungary, they are now actually rerouted because this border is closed through Slovenia, and then they want to go to Germany. So the flux of refugees per day is about like what you would have 2 million per day in the United States for a country that has only 2 million, so it's quite a huge burden, but still very handsomely managed, and so smooth operation is in place. Today I would like to talk about some other things. Endocrine, hormone released from a single vesicle. So endocrinology, of course, is a very important system. It's not as fast as it is the nervous system, but actually contributes to the homeostasis by releasing hormones into the blood. And then blood, by convection, actually distributes these hormones to different targets. To a large extent, this hormone release depends on regulated exocytosis. It's a very basic cell biological process that has been invented like 3 billion years ago, at least. So it's really a new, a very important invention that has been known for like at least 60, 70 years, but actually still poorly understood. Not only clinically relevant, but also industrially extremely relevant, because many of the new biological drugs, recombinant biology products, actually come by secretion from, in fact, clonal cells that, in fact, utilize this process for actually exiting the products, and they are synthesized. Now, if I go into further details about cell biology textbooks, you know that in these eukaryotic cells we have two kinds of vesicles that contain hormones, and these are very small ones that are mostly present in neurons, but there are larger ones that are called dense cores. Now, why there are different types of vesicles, we don't know. But it has been assumed a long time ago, 50, 60 years ago, that in fact these vesicles, irrespective of their size, they actually bear the same function. Now, what I'm going to tell you today is that in fact vesicle matters, vesicle size matters, and in fact it determines the release of hormones at the vesicle level. Now, I'm just want to highlight again, we have very small clear core vesicles, we have dense core vesicles. These ones actually contain mostly sort of peptidergic hormones and catecholamines. These ones actually contain sort of synaptic-like vesicles that contain sort of small transmitters. Now, since Bernard Katz, who won the Nobel Prize in 1970 for the quantum theory of neurotransmission, he actually, the concept of how this endocrine system works is based on this. And in fact, what he proposed, I deliberately have taken this figure legend from his book that actually has been very well read and has a very important place in neurosciences, but also in endocrinology. He actually wrote that in the nerve ending we have vesicles that contain neurotransmitter, and he wrote that these vesicles actually collide with the plasma membrane. Collision is very important in that sort of sense, and nowadays also accepted as an important sort of mechanism, and that this collision at special sites of the membrane leads to temporary membrane fusion and all or none discharge. He needed all or none discharge because of his biophysical explanation of this quantum theory. Now, let me highlight, so to understand how a molecule that is stored in the vesicle is secreted, is released in an endocrine system, so we need to have delivery of this vesicle to the plasma membrane, we need to have a formation of a hemifusion structure, and then a pore, very narrow pore that actually may enlarge reversibly, and then eventually leading to a full fusion event where actually the pore becomes so large that in fact whatever is inside can actually be discharged. Now, what I'd like to highlight here is that in fact these stages 2 and 3 are so energetically stable that in fact, when fusion of membranes occur, then in fact there is very little of the compound that actually secreted from this structure unless the pore really opens widely. Now, these experiments have been done by using a biophysical approach, on a model cell that actually comes from the anterior pituitary or the pars intermedia. As you can see here, of course, the anterior pituitary is regulated by the hypothalamic factor, blood-borne factor. They actually come and bind to each of these cell types and, of course, produces then secretion of these numerous, multiple sort of peptidergic hormones. In the pars intermedia, on the other hand, in fact, especially in rodents, but also mainly embryologically also in humans, there is innervation coming directly from the hypothalamus, and there are synapses on these cells that actually release alpha MSH and then beta endorphin. Now, of course, as a model, this is a fantastic cell type because, you know, these cells are really, really specialized to do regulated exocytosis, to release, and they do that very, very well. However, the time course of these responses are very slow. Now, to actually highlight the principle of these measurements, of membrane capacitance measurements, I'm showing here an EM micrograph of a lactotroph cell containing prolactin granules. They are really relatively large, about 100 to 200 nanometers, and whenever actually a vesicle fuses with the plasma membrane, then the area of the membrane increases by exactly the surface area of a single area of the single vesicle. Okay? So in other words, when secretion occurs, surface area, vice versa, membrane capacitance increases. So we have here a plot that actually shows area of the single cell as a function of membrane capacitance, and we see a linear relationship, which means that, you know, a unit area of the membrane stores certain charge. Now, this is like a bottle. If the bottle is small, the capacity of the bottle is small. So if a cell is small, so we have a small membrane capacitance of the membrane. If, of course, if you take the membrane capacitance of a single vesicle, this becomes infinitely smaller, okay, down to values of attofarads. I'm going to come to that later. But first, you know, we thought, okay, let's look at the whole cell. So how an endocrine cell responds in terms of membrane capacitance if we stimulate the cell rigorously. And for that, a technique has been introduced by Erwin Neher and colleagues, who actually has won the Nobel Prize together with Bert Zachman on inventing the technique, the patch clamp technique, where actually the membrane is actually reduced to actually a parallel sort of combination of a resistance and a membrane capacitance. So if you actually penetrate the cytoplasm by a pipette and introduce sort of molecules into the cytoplasm, then you can actually change calcium very abruptly. You need that because if you want to do biophysics, you need to have a step function. So that means calcium that is presumably the trigger for this fusion of vesicles with the plasma membrane should be elevated extremely fast. And this is done by actually introducing into the cytoplasm this chelator for calcium that is optically sensitive. When you flash UV light, then this molecule releases calcium, and calcium is detected fluorescently by this molecule Pheoptera. When we have done these experiments, what we have observed, like in neurons, that in fact an elevation in calcium generates a very large increase in surface area of the membrane measured as membrane capacitance. Now, this elevation is about 600 femtofarads, and if you assume that a vesicle has an area of about half a femtofarad, then this would actually mean that about 600 vesicles have been fused in a time domain of about 1 minute. If you expand this time course here, then you see that the evolution of this increase in surface area is biphasic. It starts with a very rapid increase of about 100 femtofarads, that is about 100 vesicles, and then is followed by a very slow increase to the peak of about 600 vesicles in this particular cell. Now, this is important because, you know, this approach would allow us to measure the sensitivity to calcium, and of course the release rate measured as a surface area increasing membrane capacitance. Of course, this surface area increase can be blocked completely if you would use toxins that are known to actually block membrane fusion. That is the members of the snare toxin that are botulinum neurotoxins. Now, what was interesting to see is that in fact the rapid component was very sensitive to changes in calcium, so at least at 3 micromolar calcium total, all vesicles that were sensitive to this small amplitude of calcium actually got completely released, whereas actually the larger amplitude actually required sort of much larger concentration of calcium, those that are usually present in neurons. Now, we have further developed this in a series of studies and determined that in fact in a single cell, even though we have only one hormone released, we have multiple mechanisms of release. That is, that a vesicle may contain the snare proteins that are ubiquitously present, that are the targets of the botulinum neurotoxin, tetanus toxin. In fact, these vesicles may carry other sets of proteins that determine either very rapid fusion kinetics or very slow fusion kinetics that requires very low calcium or much larger concentration of calcium. So in a single cell we have heterogeneity. Okay? Now in endocrine beta cells, in fact, this heterogeneity is likely related to the maturity of granules that are synthesized, that carry either freshly synthesized insulin or, in fact, aged insulin in these structures. So from this point on, we ask what actually determines the fusion at a single vesicle level. Okay? So we go down to nanoscale physiology, that is nanometers. And the question was, is the nature of release from a single vesicle similar to stimulated and spontaneous conditions? Going back to Bernard Katz, he predicted for his theory of release, quantum theory of release, that in fact, that under stimulated conditions and under spontaneous conditions, the nature of this fusion event is the same. The only difference is the probability of occurrence. Okay? That was his hypothesis. And so we actually aim to actually elucidate that at the single vesicle level. For this, actually, we used a couple of techniques. One is actually showing here an endocrine cell that is exposed to a dye. When you stimulate these cells, you can see that they become really bright. These granules actually that are at the confocal level in the equatorial lane just pick up this dye and then indicate the fusion pore has widely open. And in fact, they can discharge these granules that are inside. Okay? That's one approach. The other approach was to, in fact, label from the inside the granule content by using a fluorescent construct, GFP construct, that actually would target exactly to the granule matrix, and at the same time actually have the granules labeled inside and outside. And the idea would be if something is happening with stimulation, then we should see that the green fluorescence, when it's exiting, the intensity should come down. But of course, the fluorescent probe outside the cell should actually enter and then should actually label in red. In fact, this is what we have seen. Under stimulated conditions, this is a single vesicle membrane, actually is labeled red, and as a function of time, in a few seconds, the contents of this vesicle actually is diluted out, and at the same time, the dye from the outside actually emerges in. Well, the same thing can be observed under spontaneous conditions, but the time course is different. 50% of vesicles would discharge the content very, very slowly in about 16 to 20 minutes. Now, the kinetics is important, and so we can compare it again, stimulated and spontaneous conditions. The red dye comes in very rapidly, and the green dye goes out a bit more slowly, okay? Whereas actually in spontaneous conditions, this kinetics is 2 orders of magnitude slower. What is also different is the time course. So the entry is rapid, the exit is slow, and this could be related to the molecular weight. Now, in case of spontaneous release, the kinetics is not different. It seems to be synchronized. And now this is a puzzle, how to explain that. So how come a molecule exiting and entering the same structure actually has different kinetics? One would expect, if this would be a narrow tube, because molecular weights are different, there should be a distinct kinetics. But we could not actually see that. We have had to actually model this as a mechanism where actually the fusion pore opens and closes like a channel, and then we could actually make it synchronized. So this is the idea of how this should be. So if it opens and closes, and if it opens and closes like that, then we could actually make this sort of process synchronous. Now, to verify that, we decided to actually measure fluctuations in membrane surface area at a high resolution, and this is attofarads here, and these attofarads actually, if you convert it to vesicle diameter, this would be like 30 nanometer vesicle, the smallest you can see in the nervous system. Now, when you do that, you place a pipette and you start observing the fluctuation in surface area. You see this recording here. So you see just small pulses of amplitude of half a femtofarad that come very regularly. And in fact, we have 2 events, one larger and then small ones here, okay? And the lifetime of this this actually event here is about 50 milliseconds. It is not exponentially distributed, but is Gaussian, which is very, very peculiar for a system of that kind. In fact, these events come so often that it's very unlikely they are completely independent. They must be somehow linked. They really likely represent a vesicle opening and closing many, many times. Okay? Now, what is also observed here is that, in fact, we have some events that have a projection to these other trays. I'm not going to go into detail, but in biophysics, you can calculate the narrowness of this pore. And the narrowness of this spore is, in fact, such that, in fact, you can place in this neck only a few molecules of water. Okay? So very narrow, much smaller than what you would expect for the size of glutamate or acetylcholine, and of course, it's really small in comparison to a peptide hormone. Now, this periodicity, we know now it's related to some electrostatic interactions of these fusing vesicles, but the abundance of these events shows that, in fact, when a vesicle fuses, it's very stable there. And this stability actually is delineated here. So half a nanometer diameter of a pore that has been determined independently by another technique actually indicates that the pore at rest is so narrow that would not permeate even very smallest sort of hormones and neurotransmitters, and we call this unproductive exocytosis. It is, when the vesicle is fused, it's fused so strongly that, in fact, if you attach a cell to the glass cover slip and shave off the rest of the cell, the membrane alone stays, and this membrane alone would have vesicles. Now, if you load these vesicles with a hormone surrogate like alkyl natriuretic peptide and stimulate this alone strongly, you still get discharge of vesicle content. So you have a completely artificial system that operates, and of course these vesicles are stably present on the membrane alone, but also release. So the question is how you can actually make these structures so strong. So, well, in 30, 40 years ago, it was thought that, in fact, if vesicle actually approaches the membrane, okay, there are strong repulsive forces because the surface is negatively charged. So if you want to fuse them, you have to bring them very closely together, so you use a lot of energy. Okay. Therefore, it was presumed that, in fact, if you want to actually bring them very close together, then the process must be irreversible. But in fact, it's not. So this is the idea that actually is highlighted by these experiments. So we asked our colleagues, theoretical physicists, how to make a fusion pore that is narrow, so stable. And that would be the pore of this size. That would be the thickness of the membrane, that would be the pore dimension, and then again the thickness of the membrane. So the calculations show that, in fact, you can only assume the membrane extremely curved only if you have molecules of special shape, constructing the curvature. And these are called anisotropic molecules. And the model outcome predicts that, in fact, these molecules are really highly concentrated in smaller vesicles because local curvature is higher. Now, in larger vesicles, they are rare. The density of these molecules is slower, and hence what you predict is the fusion pore of these vesicles that are small, when they fuse with the membrane, is going to be extremely narrow, but the larger ones are going to have a wider pore. So this model can be directly tested. So we have data, we use this data, and we found that, in fact, the fusion pore conductance, that's the measure of the fusion pore diameter, is related to the size of the vesicles. And here we see this relationship. Now you say, okay, small vesicles, and this is the quantification here, smaller vesicles have at rest a very narrow pore. If we go to a granule that is like in pituitary cells, of course, you have at rest 1 nanometer pore diameter. 1 nanometer is very small if you consider that prolactin hormone has 5 nanometers in the shape. So basically, the pore is there, but there's no release. And you ask, what is the physiological significance by having vesicles really stably associated with the plasma membrane, establishing a pore? Well, one is that, in fact, you can make the larger pores by making the vesicles bigger. So that would be very cumbersome if you think, okay, we stimulate, for instance, with a secretagogue coming from the hypothalamus to release the discharge from the contents, and then you would have to make vesicles bigger. But that's not the case. It's too complicated. What is more likely to happen is that the relationship changes. Okay, the slope is going to be steeper. I'm not going to go into details, but this is very likely what is happening. So there are molecules that are associated with the fusion machinery, and here we have conducted experiments with the mutations against molecules that actually controls the snare complex. And what we found is that, in fact, if you perturb these interactions, you end up not with one relationship, but with two. So this is the most prevalent one. So if you interact with these molecules with snares, then you get a very narrow fusion pore, even though vesicles are large. Okay? So the question is, okay, fantastic. So we can shift the relationship under physiological conditions with proteins that are known to actually control fusion. But what has also been interesting was that, in fact, we could see larger events occurring. You know, the larger vesicles had some prevalence. More likely, we could see them. You know, in control conditions, these large events were not here. This is 6 to 10 femtofarads. So we thought maybe these molecules actually change the vesicle size. Then we conducted high-resolution microscopy on these endocrine cells. And of course, this is STED microscopy. Stefan Hell obtained the Nobel Prize last year for this. And what we could actually see with this technique is that, in fact, under control conditions and in cells that are actually exposed to a mutation, there's no difference in size. That means that, in fact, these proteins somehow control the fusibility of larger vesicles preferentially. So are these molecules that actually have been showing to you the key molecules that determine the fusibility of larger vesicles? Well, we had a very good student in the lab, and we conducted a series of experiments by using sphingosine. Sphingosine is a second messenger that is actually a lipid metabolite, cationic lipid of this structure, and it is believed that, in fact, it perturbs the snare complex. This is a vesicle here. That's the plasma membrane. So for this vesicle to actually fuse with the plasma membrane, these molecules have to pair and actually form a very strong complex. However, this synaptobrevin 2 molecule, that is the target of tetanus toxin, is not available for this unless you have sphingosine. Sphingosine somehow, removes it from the membrane, and then this molecule floats, and then of course catches the other parts of the complex, and then fusion can occur. We can see surface area, actually rate of increase of surface area increase if you put sphingosine in these cells. So she asked, Ida asked, what is going to happen if you put sphingosine and monitor fusion of a single vesicle? So she added sphingosine here, and these events are very narrow because they have a projection to this trace. And then when it become wider, then have no projection here. And of course, we can measure from the amplitude whether they are small or large. What she could see is that, in fact, with sphingosine, only small ones could be there. So anyway, she calculated how many transient fusion events, those are openings, end up in full fusion, and she found that there are about 16. And she found that, in fact, if she looked at the transient events of small vesicles and large vesicles, these are large vesicles and small, she could see that sphingosine actually would deplete the large events, okay? But of course there would be an excess of small events, as if sphingosine would push the system to full fusion, but only for larger vesicles. Now she also conducted experiments independently by labeling these granules. Once the cargo of the endocrine cell actually is released, you can put on live cells antibodies that actually attach this hormone. This would be a prolactin, and then you can actually label whatever comes out from the cell in a live cell. If these granules are large in diameter, they must come from large vesicle. So she did this, and she measured inside and outside these cores and measured them, of course, in diameter. She used super resolution microscopy, and what she found is that the number of extracellular sort of granules after stimulation increased dramatically, okay? But under control conditions, the diameter of these structures was similar. After stimulation, in fact, the intracellular sort of granules were actually smaller, as if they were containing smaller vesicles that were not able to fuse fully, whereas the diameter of the extracellular vesicles granules became bigger because they were actually released preferentially from larger vesicles. Meaning that, full fusion, complete integration of a vesicle with a plasma membrane after stimulation occurs mainly with larger vesicles. So we have a preferential sort of mechanism of regulation of hormone discharge from single vesicles based on the vesicle size. She also tested the hypothesis that, in fact, larger vesicles, that is a prolactin-containing vesicle that has also snare complex molecules like the synaptobrevin 2 or VAMP2, and she found that, in fact, if you label the same vesicle with the two antibodies, she gets a nice correlation. If she measures the intensity of signals, which actually relate to the number of molecules, she found that, in fact, in larger granules that contain prolactin, there was a lower density of synaptobrevin 2 molecules. So that was the hypothesis that may determine this preferential fusogenicity of larger vesicles. I'm going to cut short my presentation here. Do I have time still? Yes, I have time? I think I have, yeah. Anyway, the conclusion is that the intermediate stage of vesicle fusion, that is the stage between fusion pore opening that is narrow, that is widening and widens completely, is a stage that is energetically very stable. Okay? And the narrow pore that actually is established during this intermediate stage is unproductive, would not release hormone. So you may have a cell full of hormone with lots of vesicles, but because of some deficits, you may not discharge the hormone into the bloodstream. Okay? So that is one message. Then I would like to actually mention that there is a differential sensitivity of small and large vesicles. Larger vesicles tend to have a preference when there is a stimulus that they go into full fusion. Okay, so that is the other message. And of course, this is not an exclusive property of pituitary cells. We have also conducted a lot of measurements on glial cells, astrocytes in the brain that are like endocrine cells because they do secrete a number of substances into the extracellular space, and the convective system in the brain extracellular space, really transports these molecules that are over 30 or 40, including peptides. And with this, I would like to conclude, and I would like to take questions if there are any. Thank you.